more of a mixy guy. It's great. I've built up this collection of really great machines that I get very excited about. So now I run a facility where we have conventional immunohistochemistry, but now the best ultra high-plex proteomics, spatial proteomics machines, and we've been collecting all of the spatial transcriptomic platforms that people are so excited about. So I'd say actually it's an open facility. I know that you do fantastic science here, and you don't need my help. But if you did want to collaborate on panels and work and analytics, then I'd love to do that. And running these facilities requires a lot of fantastic machines, but as you know, spatial biology requires a lot of people. So sequencing is kind of easy, really. You just take some cells and you put them in a machine. But working with spatial biology is much, much harder. You need so much more work to build up the panels and to do the analysis. So I think Sabanaya was here recently, and I'm lucky she works alongside me in the facility. But we've had to bring people in to help with the analysis. And I thought I'd just share some of the issues we have with analyzing spatial biology today. So I was, again, really lucky to be involved with these folk with the NIH AMP consortium in the US. And you'll have seen this paper, I know, but I just wanted to reflect on it a little bit. So the NIH consortium was one where, you know, the Americans did not know how to do biopsies in the joints. They didn't do arthroscopy like you do, and they didn't do ultrasound guided biopsy like you do either. And they were convinced that their patients would never want to do that either, because, you know, the US is a very different place in terms of healthcare. You know, you and I, we share a very similar healthcare system. We have the NHS, you have a similar system. They're both struggling, public versus private. But in the US, it's a very different system. And they just did, I remember standing in a room full of NIH people from the US just saying, none of our patients will ever let you take a biopsy. And they were completely wrong. So we trained them how to do biopsies, and the patients did give them biopsies, actually. They were just very frightened, very scared of doing that. And so the joy of the AMP consortium was bringing together some really good quality tissue samples and actually some great minds, particularly Shoma Roy Chowdhury, so people who are really advancing in the analytics of single cell work. And so the phase 1 AMP published back in 2019 was the first single cell data in rheumatoid, as you know. And then the second phase was published just at the end of 2023. And it was originally conceived to be in 3 different clinical groups. So early disease, DMARD naive, and then methotrexate unresponsive patients, and then patients who are unresponsive to their first TNF inhibitor. And in fact, those 2 groups don't differentiate particularly in the data set. And the one thing they have in common is that they're all patients with active disease. So again, in retrospect, a better control would have been patients with inactive disease or in remission, which was really what groups in Europe were working on at the same time. But again, at the time this was built, this was an exciting tissue protocol in terms of standard H&E staining, immunofluorescence, and single cell sequencing. And as you may know, they've continued to do additional work. So they've published on some of their repertoire work now. But it's a really nicely worked out pathway for the tissue, including back up the weight of bulk RNA-seq as well, to increase the depth of the work that's done. And you'll know that because of having about sort of 350,000 cells, that gave a lot of power for annotation. So they were able to annotate a very high number of different cells. And if you're ever wondering about single cell work and wondering how to work with those data, then the best thing you can do is go and read the paper and all the supplementaries for this, and then go and work with their online facilities where you can interrogate the genes that you're interested in. The only problem with working out that there are annotating 80 different cell states is that it's very difficult to do statistics on that to understand the diversity of disease. So I guess in answer to people like Constantino Pitsalis advancing the idea of a pathotype, they wanted to do some very simple analytics. So the idea of CTAPs, or cell type abundance phenotypes, is ridiculously simple, actually. It's just looking at the relative abundance of the most crude cell descriptors, so Ts, NKs, B in plasma cells, myeloid cells, and fibroblasts. And so the idea that you might use something as simple as principal component analysis, taking all of those tissues from all of those patients and just letting them fall in an unbiased way into different groups. And I'd say, to be fair, though, we still don't really understand what this means, but we're certainly seeing relatively consistent different subtypes of disease. And the CTAPs are interesting. So one of the things that's interesting about them is that one of the ways historically we've always tried to validate observations around tissue is to try and correlate them with things like clinical descriptors. But in fact, if you correlate the variance in CTAPs against clinical measures like age, rheumatoid factor, ESR, CRP, DAS28, there's really very little correlation. So the CTAPs are correlating really, not surprisingly, with what's happening in the tissue. So the best things they correlate with are these measures of histological inflammation, the Krenn score, and there's an aggregate and density score that we designed in Birmingham. So they're not correlating with the standard indices of disease activity, and maybe that's a good thing. But if you look in the detail of the paper, you'll see that although even with the larger single cell consortium paper, there is not the power to look at individual cell clusters. Actually, those clusters do correlate quite well with the CTAPs. So you see very specific macrophage populations that are only present in the N subtype, and very specific B cell and T cell populations that are only seen in the B cell specific and T cell specific CTAPs. So it may well be that there are specific mechanisms associated with these different CTAPs. And I guess the problem with this is that we still don't have longitudinal data to look at it, but we are doing some additional work. And again, as a kind of advertising ploy for the Birmingham Tissue Analytics Service, we've done some multiplex immunohistochemistry on this, and this was published in the paper, but this is relatively simple multiplex staining. So this is only doing 7 colors at once on an Akoya phenoimager. So this was our work looking at T cell markers, macrophage markers, and then having a panel of stromal markers, because we're very fibroblast obsessed in Birmingham, while the Rochester group in New York was doing confocal microscopy with 4 markers. So it was nice to illustrate that we could do high-level staining. And it's nice to look then at the CTAPs and see that there are spatial differences between the CTAPs. So a TM CTAP, which has a high abundance of both T and M T cells and macrophages, has a very different spatial phenotype to an M macrophage, where you see enormous amounts of CD68 staining, macrophage staining throughout the tissue, whereas in the TM, really the macrophages are just sat in the sublining area. And really understanding more of those spatial relationships is the next step for this tissue. So in terms of our work for this, we're going to use this PhenoCycler platform, which allows you to look at up to 100 markers. And you may have heard about this platform. We run 3 different high-plex platforms in Birmingham. This is a nice one that was developed, in Stanford by Gary Nolan. So the idea that you'd have iterative staining, so you stain for 4 colors at once on the tissue. How do you do that? You do that repeatedly, and you do that by using oligonucleotides. So you have fluorescently labeled oligonucleotides that are complementary and that are linked to each antibody. So you can put on 4 different antibodies every time, but you're picking up those new 4 antibodies with new complementary oligonucleotides. So it's a really nice fusion of proteomics and using RNA technology to specifically label those. So Akoya themselves will advertise their technology with this 101 antibody panel, which must have taken them a long time and a lot of money to put together. But you can see what they're able to do. You can see the number of markers they're able to look at. If you look at 100 markers in cancer tissue, that's quite exciting. And then they can have their very exciting diagram where you have the spatial data segueing directly into a UMAP of the different cellular populations. So it's visually very attractive, but it's definitely very useful in terms of understanding those spatial arrangements. So I can't show you the final deal on the AMP at the moment, but the PhenoCycler platform is looking very, very nice. So this is some rheumatoid arthritis tissue, and these are some of your familiar markers that you'll understand as well. In this case, we're looking at T cell populations, trying to pick out Treg cells, FOXP3 positive staining. But you can see this really beautiful staining of the stromal layer, podoplanin positive fibroblast interdigitating through the lining layer. So the sort of resolution we're getting on this platform is really nice, actually. So it's much better quality than the previous ultra high parameter platforms that we've used. And so that's going to be really informative. Again, this is looking at an aggregate a tertiary lymphoid aggregate in rheumatoid arthritis. And again, you can see really nice delineation of the cells, and the magnification is good enough that we're going to be able to annotate quite well within aggregates, which is always a problem. Okay, so end of that story. Rheumatoid heterogeneity, which you're more experts in than I am, really, certainly broader than simple pathotypes, but it's the longitudinal tissue studies that we're going to need to understand what the meaning of CTAPs is. So checkpoint-induced disease, you must have this new speciality too, mustn't you? So patients with cancer are given immunotherapy, they're given checkpoint inhibitor therapy, and then we've created this amazing new iatrogenic. I mean, how much of your work, I wonder, does this take up clinically? It's an increasing part of our work. Yeah, so, and I'm sure you've been sampling those patients as well, haven't you? So you may well have your own data on these patients as well. And we started looking at these patients at the same time as some of our colleagues in the UK. We have a consortium who work very closely together in Birmingham, Newcastle, Glasgow, Ian McInnes's unit in Oxford. And so we were trying to work out what exactly was happening in these patients. And we took a slightly messy approach to this. So again, you'll be aware of this idea about immunotherapy, that normally the T cell responses to tumors are blunted because the tumor cells express PD-L1, and that then engages with the checkpoint inhibitor, the PD-1 checkpoint inhibitor on T cells, so the tumor can escape immune evasion. And so obviously our oncology colleagues had this great idea that they'd inhibit that conversation. And as you know, then inhibiting that or blocking that inhibitory checkpoint inhibitor then leads to hyperactivity of T cells. And so again, about 6% of patients who are on checkpoint inhibitors will develop an inflammatory arthritis. And you must have noticed as well that unfortunately the arthritis doesn't usually resolve. So the majority of patients, the arthritis continues. So our messy approach to this was, and it probably is messy, is to look at different populations of disease. So we have a clear-cut, quite well phenotyped population of seropositive rheumatoid patients. We have some psoriatic arthritis patients, but we also have this sort of messy population that we see of seronegative inflammatory arthritis that includes patients with kind of polymyalgia rheumatica overlaps. They're an underserved population, they're not well described. And phenotypically, they seem to resemble a slightly messy population that we get with immune checkpoint inhibitor-induced arthritis. So this was really a simplistic hypothesis, was that we thought we might see molecular similarities between the seronegative arthritis and the immune checkpoint inhibitor that might differ between the more clearly defined seropositive rheumatoid and psoriatic arthritis. So the numbers in those cohorts are not huge, but they're probably large enough to do some meaningful work on. And so for this, we were putting together resources with Arthur Pratt. And he has a really nice prospective cohort in Newcastle where they take all the immune checkpoint inhibitor patients and they follow them over time. So they have a longitudinal study at baseline. And then they can pull out patients who do arthritis, thyroid disease, Addison's disease. So that's quite a nice cohort. But in this case, we're just looking at the arthritis patients. This is my colleague, Ben Fisher, who's a great trials designer and a clinical trialist in rheumatology, which I'll show you a bit more of in a minute. But he and I were working together looking at these immune checkpoint patients. So the approach that we've taken to this was classical, what is now a classical approach of single cell transcriptomics. And we've also been using the spatial transcriptomics to look at this in more detail. So it's quite a big team approach to look at this. And I think although it's super exciting to show data on spatial transcriptomics, and I'm sure you're doing this too, as you may know, the problem with this is actually the analysis. That's the tricky area, in that things like the nanostring and the 10x machines come with some analytics with them, but it's not great. They're not fully worked through. So you have to evolve your own approaches to analyze that. If we just look at the single cell data that pop out of this, then again, we have this kind of 4 groups of patients that we're looking at, and just in a crude way, in a sort of CTAP sort of way, where we're looking at just the different simple cellular groupings. What you see is that the checkpoint inhibitor-induced arthritis sit fairly squarely in either the fibroblast dominant grouping or the T cell dominant grouping. You see very few of them in this very macrophage-driven grouping, and actually the diseases that occupy that classical myeloid driven disease are seropositive rheumatoid arthritis and PSA. So the checkpoint inhibitor-induced disease does seem to have some sort of phenotypic characteristic. And when you look at the different fibroblast populations, so on the bottom, you've got a graph here, a chart of fibroblasts, unspecified fibroblast cells from across the groupings. And on top, you've got mural stromal cells. So you do see a predominance of stromal cells in the patients with checkpoint inhibitor-induced arthritis. And there are other differences as well. So you probably looked at the tissue from these patients as well, and you often see really florid neovascularization. So this is just one of the endothelial subtypes. We see a lot of new vessel formation in checkpoint inhibitor-induced arthritis. And the other thing you see prominence of are CD8 T cells. So I said there were a T cell predominant phenotype we see in the tissue, and sure enough you do. So some of that overlaps with the other groups, but you always see high levels of activated CD8 T cells and this grouping of granzyme K positive, CD8 positive T cells that have become the subject of recent interest. And this work's been done by Jason Turner, who works with me, who's an informatician, and Anthony McLean, who's his opposite number in Newcastle, working collaboratively. So in terms of mechanisms, the thing that we got excited about was, oddly enough, even though macrophages aren't predominant in these tissues, what we saw quite consistently across the different tissue groupings was an MHC1 positive gene signature. So the macrophages in the non-seropositive rheumatoid arthritis are very strongly biased towards MHC positive antigen, MHC class 1 antigen presentation. And they also express CXCL9, 10, 11 very positively. So it's quite a specific population of macrophages. So they're expressing interferon gamma driven chemokines. And the receptor for that, for those chemokines, CXCL3, is very strongly expressed by those CD8 T cells that we see accumulating in disease as well. So we have an interesting story where we think we're recruiting CD8 T cells towards these macrophages. And what I'm not showing the data for here, but what's very clearly shown in expression as well, is that those macrophages are strongly expressing PD-L1. So the ligand for the checkpoint inhibitor. So on that original diagram I showed, where you have an antigen presenting cell or a cell switching off immune responses, instead of a tumor cell here, we've got a macrophage expressing a checkpoint inhibitor ligand, and the T cells normally would express that PD-1. So if you imagine in the absence of a checkpoint inhibitor, this is a nice inhibitory pathway. So when there's no checkpoint inhibitor present, the macrophages are switching off the T cells and the T cells are quite quiet. But when you interrupt that, what you then end up with is a freedom and a release and activation of the macrophages and of the T cells as well. So we think this is a key mechanism going on in these patients. And coincidentally, recently as well, the group at the Broad and the AMP consortium were looking at exactly these macrophages and did some really nice work. Actually, they were reviewing multiple tissue subtypes, tissue samples that came in during the COVID pandemic, and they'd noted these interferon gamma driven CXCL9, 10, 11 positive macrophages as being very strongly TNF and interferon gamma driven. So not only do we have this population here, we were able to interrogate that a little bit more. And you see that indeed in our cohort as well, those cells are very strongly TNF driven, but particularly in the checkpoint inhibitor induced arthritis, those macrophages show a really strong interferon gamma drive. So that fits with our observations and suggests that, again, once that checkpoint inhibitor has been used, it really unlocks a lot of interferon gamma signaling. So this is our current hypothesis. It makes sense intuitively that rheumatoid arthritis is a class 2 driven disease. These are a class 1 driven disease, but the checkpoint inhibitor induced patients are particularly interesting. So I'll just share a little bit of the work we've been doing with the Xenium. So this is a single cell resolution spatial transcriptomics machine, and we too now get to have our funky diagram where we show the spatial annotation of the cells and a UMAP. So this is using RNA expression by the cells. And this is a sort of image we get out. So it's a classical image of synovial tissue, but now the staining that we're getting obviously is RNA species rather than rather than classical antibodies. But under the microscope, these still look relatively similar. We can still see, in this case, the RNA species being identified with the platform. And I'd say that the biggest challenge, as you probably know, with these platforms is still segmenting. I mean, segmentation of cells is always the biggest challenge anyway, in cellular work and in tissue. But using this particular RNA species where it's very dotty, very sparse populations, the biggest challenge is to annotate the cells accurately, and that's going to remain a challenge for the time to come. It works in a reasonably satisfactory way. So we're able to annotate, again, broad cell types, just as we did in the single cell data. That's nice. And then we use Banksy, which is interestingly named, but it's a nice tissue annotation algorithm for picking out neighborhoods of cells. And it looks not just at the cells that are present, but also at the RNA species that they express as well. So it's a hybrid of those two things. And it just, in an unbiased way, identifies different neighborhoods. But it's neighborhood number 2 that we were interested in here, which is very much the center of aggregates. And if you look inside that number 2 neighborhood, then what you see is this predominance of, in the first place, T cells and macrophages, which is nice. But if you then look at the finer clustering, you're beginning to see, not surprisingly, not only CD4 cells that you'd expressed in an aggregate, but you're seeing CD8 cells as well. And in particular, you're seeing this population of macrophages. So it's really nice that that spatial relationship is really tight, actually, and it seems to exist around our old friends, the aggregate. And again, the xenium is nice. You can still go back in, you can look at those cellular populations. So here we're looking at TNF expression, so a driving cytokine expression or RNA for it. And we're looking at the expression of those chemokines. And you can see that they're broadly correlated. So we're expecting cells, we're expecting those two to not be expressed in the same cells, but to be expressed in neighborhood cells. And we can look at that using nebulosa plots. So we can look at expression of our key factors here. So what we're seeing here is the expression of CXCL9 by macrophages, CXCL3 by complementary CD8 T cells, the driving cytokines present, TNF and interferon gamma present at the same time. And I mentioned the fact that the checkpoint inhibitor and its ligand is also present in those cells. So you can see a coincidence of all those things being expressed at very high levels within the immune aggregate. And again, there's various other ways we can look at this, but this is applying the same sort of work we'd use with proteomics, but applying it to RNA species instead. So you can see that in the checkpoint inhibitor-induced arthritis and the PSA, and this is a log expression here, this is distance from the aggregate cells. What you find is that in checkpoint inhibitor and psoriatic disease, there's a very steep rise in CXCL9 expression as you get towards those cells. So they're really hyper-expressing that chemokine. So what I've shown you here is that those synovial are fibroblast and T-cell enriched in checkpoint inhibitor-induced arthritis. They are highly vascularized, and we think that what's happening around those perivascular aggregates is that the specialized population of macrophages are recruiting CD8 T cells and driving that very active disease that we see in checkpoint inhibitor-induced disease. And certainly the most obvious thing you'd want to do with those patients is use blockade with TNF and possibly JAK inhibitors. I'm sure you're probably already using TNF inhibitors in these patients. Ben was lucky to get funding for a formal clinical trial. So we're trialing TNF inhibitors against standard of care with steroids at the moment. TNF is already becoming standard of care, and as you'll know, there's all sorts of issues around what sort of treatment we should use with these patients and whether their patients, whether using biologics is going to interfere with their cancer therapy. In the first instance, then we're using adalimumab in this trial, but this is a platform structure. So obviously looking at something that inhibits the interferon gamma axis, particularly JAK inhibitors, is probably going to be the next step for this platform trial. Okay, so the last thing I was going to do is take you to Paris. It should be a trip to Lisbon. Apologies for that. But it's a trip to Paris because that was the name we chose for a clinical trial. So I guess, again, I wanted to highlight the work that we do with tissue in Birmingham. So for a while now, we've been working with industry with Johnson & Johnson And that came out of this work. So again, we were quite lucky to have engaged with the grouping who became the AMP consortium. So Deepak Rao works in Shoma Rochaudhry's lab and with Michael Brenner in Harvard. And we were doing this tissue work with them where we were looking at proximity and the presence of different tissues, doing again some tissue staining. This is our tissue staining work. And what Deepak had noticed was the presence of these PD-1 high T cells, so different population, but again expressing our favorite checkpoint inhibitor. And these PD-1 high T cells were being seen in seropositive rheumatoid arthritis, and they did some really nice mechanistic work showing that these T cells, these T peripheral helper cells, were driving B cell responses, which of course are very important in rheumatoid arthritis. So they described the phenotype of these cells really nicely. PD-1 high, CXCL5 high, unlike TFH cells that we normally see around lymphoid aggregates, which are CXCL5 low. So they look like active cells that are driving disease. That was back in 2017, and actually the Paris trial should have happened earlier, but we were interacting with various different industry partners who were developing PD-1 agonists, and you'll have seen the results of some of the PD-1 agonist trials. They're now coming through into the public space, and some of those trials are looking quite promising for rheumatoid arthritis. So with J&J, when we eventually worked out they had a nice PD-1 agonist, in this case it's an agonist depleter antibody, so it agonizes PD-1, so it hopefully switches off those T cells, but it also depletes very high expressing T cells as well. And what we wanted to do was look at the trial concept in a more advanced way. So we looked across 3 different tissues, initially in tissue explant studies, and then eventually designed a trial, a basket trial, around tissue outcomes. So we designed a trial in rheumatoid arthritis, Sjögren's syndrome, and ulcerative colitis at the same time, and all of those patients were given a PD-1 agonist drug. They were biopsied at baseline, and then they were biopsied at 12 weeks after. So the classic design that you guys evolved over time and that's always been used. But we're doing it across three treatments at the same time. And you're familiar with these techniques, so I won't tell you anything about ultrasound guided biopsy, except we still use this old-fashioned technique. We still use portals and forceps because we like the quality of tissue we get out of those. But we also use tissue biopsy needles the same way you do. So the primary outcome measure of the Paris trial was calculated using single cell sequencing and site-seq. So it wasn't flow cytometry, it wasn't a clinical outcome, it was actually looking in the tissue to see whether the antibody was doing its job and depleting T cells. And so from that, we got all of the wealth of data that you get from single cell sequencing. So actually, I don't have all that data to show. Well, I do have the data, but I don't have the final analysis to show you because there's going to be a lot of information that will come out of this. But assembling those data did involve quite a lot of collaboration in Birmingham and making sure that we were very good at creating these trial outcomes. That's a bit irrelevant, isn't it? But we also did some tissue staining. So this is Sabanaya, who you'll know from her work on Sjögren's as well. So we got very good at staining the rheumatoid arthritis tissue, the salivary gland tissue from Sjögren's patients, and the colonic tissue as well in our facility. And obviously we had panels illustrating the expression of PD-1 and their complementary ligands. And so we're particularly interested in the effect that the drug would have on depleting PD-1 positive cells, but also interested in whether that promoted the presence of Treg cells as well. So these are the simple outcomes of that trial. And the easiest thing to say is that actually ulcerative colitis started off as a viable cohort, but very rapidly had to be stopped. Those patients didn't respond at all. And because it's ulcerative colitis and those patients get very sick, it was terminated after the first 3 or 4 patients. So we do actually have about 5 patients altogether with samples, and 3 of those had pre and post. But you may have seen just in the last few weeks, somebody's just published the other PD-1 agonists totally failed in inflammatory bowel disease. So we already knew that it didn't work. But actually at a tissue level, in Sjögren's disease as well, our primary outcome, so this is single cell reported outcome looking at the impact of TFH and TPH cells of the drug, there was no change in Sjogren's disease. And in fact, none of the other outcomes for Sjogren's disease worked. And you might say, oh, that's a disaster. You know, the drug hasn't worked. But actually, what's fun about this and what's interesting is that you've then got the comparator of a drug that didn't work in one disease and did work in another. So in rheumatoid arthritis, we definitely got depletion of those PD-1 positive cells. And more than that, we could look at other indices as well. So interferon gamma positive, CD4 positive T cells, so activated T cells were also depleted. Peripheral CXCL13, which is a marker of aggregate formation and disease activity in Sjogren's disease and rheumatoid arthritis also fell. CRP levels fell in the rheumatoid arthritis patients. Their DAS28 levels fell. And you can correlate that change in the PD-1 positive cells positively with a change in DAS28. They're subtle effects. They're not enormous, but they all move in the same direction. And looking in the tissue, then you can look at staining for T cells, which dropped, staining for B cells, which we think are being helped by the T cells, which also dropped. And even things like the Kren score also dropped over that time. So everything moves together. And the interesting thing then is to be able to look back at this data. So we can look at this data, Johnson & Johnson can look at the data, and we can say, well, not only did the drug not work in Sjogren's disease, but we can say why it didn't work in Sjogren's disease. It's not hitting the cells it should, it's not depleting the cells that it should do. And then we can start to form hypothesis about why that didn't work. Is it that we need a higher dose of the drug in Sjogren's disease, or is it just that it'll never work at all? So really the purpose of this was to show you that we think that tissue-derived outcomes are the way to go in clinical trials. And that's going to allow us to make very early decisions. It's going to allow us to bring drugs forward more quickly. We can give the pharma companies the information that they want very quickly, but we can also find out more about the mechanisms of action of the drugs, and we can find more about the biology of disease as well by understanding how drugs work in clinical trials. And we're actively moving both single cell and advanced spatial analytics into the clinical trial space. And again, we're very excited to collaborate with partners who are doing that so that we start to apply this level of rigor to routine clinical trials. So really my final slide was more thinking and discussion, really. My experience of spatial platforms is that they're fantastic, but actually they're very expensive to operate. It's difficult to persuade your university you need all those people because it's incredibly expensive. It's expensive work to do, but it's very, very valuable work. It is the next generation. So although the truth is in the tissue, which is something that my mentor, Chris Buckley, would always say, but actually the future is absolutely spatial. Those spatial relationships are really what allow you to validate and understand what's happening in disease. And we're going to bring those things into clinical trials as quickly as we can. But actually the biggest issue is that the analytic platforms often are being brought to market so quickly that they're often not fully formed. And so the biggest challenge we have is having the people and deriving our own ways to understand those data that we get out of the spatial platforms and to analyze them. So I hope that didn't confuse you too much, but it's the result of a lot of work through different groups, particularly in the UK, where we collaborate with Newcastle in a big way and Glasgow as well. So thanks for your attention. Coffee sounds good.
I was wondering if the special localization that you have for interferon gamma cell plus the macrophage that you see around have any sign of having a kind of exclusion zone for M2 like macrophages. So around, see my point? So
yes,
you show this type of phenotype of macrophage that I assume
M1 like.
M1 like.
Yeah.
M1
Yeah.
Have you observed a kind of an exclusion zone for the M2?
That is a beautiful question that I don't know the answer to, but I can find out. So that's great. Mm.
not a special. Maybe that's something. Another point is mentioned the interferon gamma and the development of in PD. Probably interferon gamma came across with them.
I think so, and the closest thing we'll get to that we can use that's already standard of care is a JAK inhibitor, I guess. But a direct interferon gamma inhibitor is definitely something to use in that group, as long as it's safe in the cancer population. Yeah. No, I mean that's fascinating, isn't it? So you could use lymph nodes, couldn't you, from all of the patients? Yeah. 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 No, that's yeah. And also, yeah, and colon tissue, yeah. So it's not a direct comparison across the same tissue. I mean, that would be a very elegant but very difficult study to run, wouldn't it? But I see your point that would be the perfect control, wouldn't it? Yeah. Well, in a way you can't, I guess. I mean, you're looking at the point where you want the drug. What you're doing is looking at the point where you want the drug to act, I guess. So you do have to do some very careful work first with the single cell, for instance, in terms of how you digest the tissue. So you digest the tissue in the same way. So you're looking at the cells and you're looking at the cells and the tissue in the same way. You're not biasing because of the way you select, because of the way you process the tissue. Right. No, I take your point. The perfect control will be to do every tissue in every patient. Well, because I guess the power is that because it's a longitudinal study, isn't it? So you have the power of each individual patient, that you've got a baseline and then you've got a longitudinal measurement. So that in itself is what you need to say whether there's a change. But you're right that you can't. So again, having the single cell data in the tissue then allows you to infer mechanism, but the mechanisms could be completely different in each tissue. So the fact that there was no response in Sjögren's disease, the interesting thing about that is that at baseline, actually their absolute levels of PD-1 positive cells are much higher. So it may simply be a dose issue. In other words, you're depleting PD-1, but you're not depleting enough PD-1 to have an effect. So we have those other pieces of information. We have the pharmacokinetic data as well. So we can put those things together. But that allows you to understand so much more about why a drug is working or not working when you do the original study. I think if you wanted to match, ideally, maybe you'd try and find a lymph node, but a mesenteric lymph node is going to be difficult to sample. But we're certainly starting to do studies where we're looking in Sjögren's, we're looking in the skin and the salivary gland and the synovium, and we're taking lymph nodes at the same time to understand in one common tissue. So we're running a trial like that at the moment. Yeah. Yeah. It's tempting, isn't it? Because they all overlap, don't they? You do get joint involvement in Sjögren's, you do get. So Rita, I think you need to talk to Chris Buckley, my old mentor, because he wants to biopsy every tissue in every patient at every time point. I know why he wants to do that, but sometimes you have to be pragmatic. It is the dream. I think the lymph node is the key for a lot of those things, isn't it? To understand what's happening with the adaptive immune system. If you can access the lymph node, then you have a common tissue and you can look at common effects. And that in that environment, PD-1 positive cells that actually would be a great tissue to access. Joachim.
How are we struggling to personalize the treatment? We are trying with the peptides, now with the CPEP, single cell, we are in more detail in tissue, but we are still struggling to personalize the size and each treatment for each patient. We think that part of it is because we are selecting the patients for the biopsies. We use the algorithms from computer more accessible, but the heterogeneity that comes from that, how can we overcome it? And what are we missing our objective?
I don't think we need to search for complicated things. It's just power. We're rheumatologists and we like working in the grey, don't we? We like working with heterogeneity, but the trouble is we've chosen an incredibly heterogeneous disease. So rheumatoid arthritis is a syndrome. It's a ridiculously heterogeneous, compared to ulcerative colitis and Crohn's disease, which have an element of pathology in their diagnosis. Sjogren's formerly, has an element of pathology in its diagnosis. Rheumatoid does not. So we've given ourselves the hardest task of several diseases. So not just several CTABs, but several diseases. And we just have to embrace that. So although we spend time talking about, oh God, I wish we could do what Stefano's doing in Rome. Because in our health services, we can't do what he's doing. So he's systematically biopsying every patient that comes through the door, which is amazing. But through doing that, we will get a handle on the heterogeneity. And the trouble is, it's a numbers thing. So the more we join together and do studies together and increase our numbers, the more we're going to understand about that. And the checkpoint inhibitor disease is interesting because that is more homogeneous, actually. When you look at those signatures that come out, they're really strong signatures. The rheumatoid is just very difficult. So, Costantino's advancing with that, isn't he? He's got gene signatures now that predict response, but actually those studies need to be done in about 5 times more patients. You need about 1,000 patients on biologics before you can understand the relationship between heterogeneity and response to drugs. So we've set ourselves a really difficult challenge. But what we have to do is do a lot of work together. So I haven't talked about it, but I have a pre and post biologics cohort, and I bet you do too. I bet you're collecting tissue from patients just on standard of care drugs. Stefano is as well. So what we should be doing really is as long as we're collecting the data in a robust way, we should be putting those together. That's why AMP worked, is because they actually got 80 tissues and processed them all at the same time in the same way. All of the other cohorts, shamefully, my checkpoint inhibitor arthritis cohort is too small. You need bigger numbers to understand heterogeneity. So there's no mystery. It's just you've got to be big. You've got to think big. More biopsy. Any more for any more? So we think not, don't we? So I didn't show it, but in the AMP paper, they didn't have any longitudinal samples. So they had to go to Costantino. So they got the CTAPs and they used CyberSort and projected the CTAPs onto bulk data that Costantino has. And they showed that about 30% of the CTAPs are changing after therapy. So, oh my God, it's almost time to restart. No, not in the middle of this. So I think they probably do change. I don't think patients have completely robust CTAPs. And that's the other problem, is that they're probably dynamic. And until we have enough longitudinal studies. So not only do we have heterogeneity, we have change over time. So you know what that means is that we all have to do longitudinal cohorts and we all have to put our results together and we all have to do the same type of sequencing on them. The only good thing is that all of the informatics to do that are accelerating rapidly and are good enough to do that. So actually all we need to do is get together and discuss what tissue samples we have and make sure that we collect data in a good enough way and that we can put those two things together. It's really, as you say, it's really tough. They're definitely not stable CTAS, but my longitudinal, it's going to be, you know, I'm going to have longitudinal data on a lot of patients, about the size of the AMP cohort, and Stefano has something similar. If we put those together, we should be able to get a really powerful handle on it. If you imagine you have, you know, single cell data on 500 patients, that would start to answer that question, but you need those numbers to answer that question. Massive cost, massive cost. And the trouble is, it is the sort of thing that funders, it's too big for most funders because of the costs involved. So you have to do your own work and then find a way to put those together across academic institutions, don't you? Frustratingly. It doesn't help that, you know, the perception is that rheumatoid arthritis is solved, isn't it? In some quarters that we've fixed rheumatoid. We know that we haven't. We know that, you know, we've just created patients now with refractory disease. So we've just created a different type of patient now. Always challenges. Oh man. Oh.
Difference in stimulus, less osteoclast activation.
I guess we can only talk about what we have, and we won't have tissues that are rich in osteoclasts. We probably don't have those cells in our data sets. So we'll have a lot of macrophage populations. We won't have osteoclasts. But if you wanted to get together with us and talk about those different populations and do some collaboration, and we look at the different myeloid populations, then we could look at those together. But we won't have osteoclasts, and we'll only have precursors. No, I haven't checked. Yeah, this is kind of ongoing work rather than packaged up work. It's a nice question. Yeah, so if we're talking about comparing those two, I couldn't give you, I'd have to look at that analysis. I don't have that at my fingertips, actually. I don't think there's much of a difference between the two. So the biggest difference histologically is in the endothelium. So if you're talking about lining layer hypertrophy, there's no difference between those groups, really. But I think yeah, exactly, synovitis, isn't it? But it's definitely a different flavor. I mean, the vascularity is obvious to the eye, really. So that's an interesting thing to pursue. That's another area where artificial intelligence is nice, to start looking at regional differences. You look at something and you think, oh, it looks a bit different. But actually, if you start to use AI to recognize regional differences, that can sometimes pull out differences that you can't systematically recognize. So we're doing some work on that. Nice questions. Oh, thanks.